Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a part of the team here at SynGAP Research Fund. Our presentation today is Perturbations of SynGAP1 Function by Coding Sequence Mutation and Development of Automated Methods for their Quantitation. I have the pleasure to introduce today's speaker, Dr. Michael Courtney. Dr. Courtney leads the Neuronal Signal Lab at the Turku Bioscience Center in Finland. He also established and is head of the Turku Screening Unit, a facility affiliated with Finnish and European infrastructures for chemical biology and early-stage drug discovery. The unit offers access to lab automation instrumentation and provides services, in particular for use of live cell high-throughput microscopy, as well as computational screening approaches from partners in structural biology teams. Dr. Courtney completed his PhD at the University of Dundee in 1992 and moved to Finland to develop optical methods to study neuronal cell signaling. Such methods continue to develop rapidly and are becoming increasingly important to identify disease mechanisms. They can also be used in phenotypic screens to help identify drugs and other compounds that may have the capacity to restore specific cellular functions. This talk will describe recent work to develop miniaturized live cell optical methods to help streamline the phenotypic identification of functional defects in variants of SynGAP1 and their application to variants known or suspected to cause disease. This work is funded by a joint grant from SynGAP Research Fund US, SRF Europe, and Lyon in France. A recorded version of this webinar will be available on the SRF website under the Resource tab. And by the end of this presentation, you will have the opportunity to get the answer to your questions, and we'd love to hear from you, so please write your questions in the Q&A below. For those just joining us, welcome. And again, our speaker today is Dr. Michael Courtney to discuss perturbations of SynGAP1 function by coding sequence mutation and development of automated methods for their quantitation. It's now my pleasure to turn things over to Dr. Courtney. Thank you.
Okay, thank you very much, Olga, for that introduction, and thank you to SRF to give me an opportunity once again to tell about our work related to SynGAP. So I'm sharing my screen now. Can you see that?
Yes.
You see the slides?
Mm-hmm.
Yep, great. Okay. So let's see. Okay, good. So what I'd like to cover in this presentation is kind of to explain what it is that we're working on in relation to SynGAP1 and why we are taking the approaches that we are. And as Olga pointed out, this is a ongoing project funded by SRF and Lyon and Friends and focused on missense variants of SynGAP1. We also have a new Horizon Europe project on neurological disorders related to RAS, which includes SynGAP, which is a large 15-partner consortium project. I'm not going to talk about that today. It's just starting. But for this presentation, I'd like to introduce some concepts that are important for what I'm going to try and present, and then to explain how we're trying to efficiently measure SynGAP1 function and to determine whether these measures that we are obtaining are robust enough for future restorative drug screens that could be performed. So I think this community is quite familiar with SynGAP1 disease, obviously. So what I'd like to kind of skip some of these basics and get to the point here that missense variants are currently categorized in ClinVar as pathogenic or likely pathogenic, benign or likely benign, and of uncertain significance. So one of the questions that arises is how are these missense variants categorized, for example, as pathogenic? And I think this is based primarily on a prediction because we know quite a lot about protein structure. On the other hand, there's a lot we still don't know about protein structure. So this is a representation of a structure of SynGAP from AlphaFold. It's maybe not the very best representation, but it's considered to be not bad. And the color coding here is showing how confident we are about the positions of different parts of the SynGAP1 protein. So the dark blue and light blue shows a pretty high level of confidence of the average positions of amino acids forming the protein, particularly in this region, which has various well-known globular domains, pH domain, C2 domain, and gap domain, and also this coiled helix. Now, in the particular case of SynGAP, this isn't just SynGAP1, this isn't just a prediction, it actually is supported also by crystallographic data. This is data obtained from a crystal of the purified protein, which basically confirms these blue regions. Having said that, this is a very static view of the protein, but in a cell, the protein is very dynamic. There's a lot of flexibility, and this is not captured either by this model nor by the crystal structure. Or in fact, the crystal structure indicates flexibility and dynamics by not being visible in certain parts of the structure. But what I'd like to highlight here is all these orange and yellow regions where the confidence is extremely low. It means we really don't know what this part of the sequence is, where it is positioned, what kind of forms it may take, and it may very well be quite unstructured, which means that it's very flexible, it can take different conformations. It may have, it's often the case that these unstructured regions form specific interactions with partners, in which case they then become structured in the presence of the partners. But if we don't know what the partners are, we won't know what their structures are. So we really don't have a lot of information about this, the rest of the sequence. It kind of looks like spaghetti here, which means that the prediction is very poor in these regions. So if we don't have the possibility to predict everything about the structure just based on the sequence, then perhaps we need to look at experimental evidence. And this is what the focus of this presentation will be. What kind of experimental evidence can we get about what is the consequence of a missense variance in the SynGAP sequence? And is this evidence useful for other things, specifically for drug discovery? Okay, so another question that arises is whether all pathogenic missense variants are acting via haploinsufficiency. So in the case of the most common known forms of SynGAP, there is a nonsense mutation. That means an early stop codon in the coding DNA and thus the RNA produced from the DNA in one allele, one gene copy of SynGAP1. And there are mechanisms in cells called nonsense-mediated decay, which are recognizing these early stop codons and destroying messenger RNA that contains these early stop codons. And the consequence of this is usually a protective mechanism to make sure that fragments of proteins and half proteins aren't produced, because they may interfere with the function of the normal full-length protein, which is encoded on the other of the two copies that we have of every gene. And unfortunately, SynGAP1 is one of those genes where a single copy is insufficient for function. And that's why it's called haploinsufficiency. So the second copy is described as loss of function. So that's very clear in the case of these nonsense mutations. But when it comes to SynGAP missense variants, the question is, does this substitution, I mean, there are some parts of the protein where if you take out one amino acid and put in another one instead, the whole structure will become unstable. And there are again mechanisms in the cell that will, in this case, degrade an unstable unfolded protein. So we may just have no protein again. So this is clearly a loss of function, like a haploinsufficiency, like the nonsense mutation. On the other hand, it may be that some missense variants lead to a protein that still can't fold, but perhaps it's exposing some hydrophobic regions because of this failure to fold. And these hydrophobic regions can form aggregates. And there are many neurological disorders where protein aggregates are known to play a role. So this could be one of the outcomes of a given missense variation. It could be, on the other hand, that there is expression and there's solubility of the SynGAP protein, but it's simply in the wrong place. SynGAP should, in part, be at the synapse. And if it doesn't get to the synapse, there will be some problems. On the other hand, it could be at the synapse, but unable to act on its targets, or perhaps it can act on its targets, but it doesn't respond to inputs. There's a lot of different reasons why the missense variation could lead to disturbed or loss of function. In each case, we'd have to take a look to kind of determine what is the issue. And then there's a different, kind of conceptually different consequence that's possible from a missense variant if it is expressed but not functional, because it could take the place of the wild type protein produced from the wild type allele. And this is called a dominant negative function, where it could be a simple displacement where at the synapse, for example, there may be a missense rather than the wild type, and therefore the activity will not be present as it should be. On the other hand, SynGAP has this special property to form trimers, and it may be that substitution of just one of the three in the trimer of wild type with a missense could actually poison the entire oligomer. But we actually don't really know enough about these trimers to really be sure about that. So that's a bit speculation at this time, but it's something to investigate. Okay, so why does all of this matter? Well, if a missense mutant really does act as dominant negative, this is a problem if we now think about the therapies currently under development to counter haploinsufficiency, because these are designed to boost expression of SynGAP protein from the genes. This means the normal wild type allele, and it also means the variant allele. So if the variant allele is actually a dominant negative suppressing the activity of the wild type, then these kind of therapies may even be harmful, in which case the missense SynGAP, those with missense SynGAP will be excluded from the trials and won't be able to get any benefit in the steps for their own safety. However, if we can determine what is wrong with the missense variant and actually put numbers on it and quantify it in comparison with the wild type, it means we could potentially set up a screen for compounds to try and find compounds which make the variant more like the wild type. So a word about compound screening, there are basically two different kinds of compound screening. So big pharma tend to go for this screening of novel chemical entities. And in this case, they go for very large libraries. They often hold libraries of more than 1 million compounds that can be tested one by one. On the other hand, there are these resources like DNA-encoded libraries that allow a pooled screen of more than 1 billion different chemical entities. And the idea here is to find a number of hits, which themselves are never going to be drugs, but they can be optimized, and medicinal chemistry can be applied, various safety studies need to be applied. It's a very long and expensive process, but in the end, a novel compound may come out of it that can be used as a drug in clinical trials, which itself poses additional challenges for rare diseases if there are not all that many known patients with the disease. So this is not really what we're talking about in this case. On the other hand, what we can do is instead of searching a novel chemical entity, we can search for existing drugs. This can be either agents launched for treating other diseases, or at least have been in a clinical trial. Because for these compounds, there is already safety information, there's already clinical data. So it's already in principle possible and safe to, or given certain conditions, for someone to take these compounds under guidance of a clinician. So in this case, the number of compounds is way smaller. We're talking about something like 4,000 or 5,000 at most that can be screened. There are still regulatory challenges. There are still costs for phase 3 trials. And the big problem here is that these drugs are already patented for one or another purpose. Some of them are, the patents are expired. So the IP potential is very low in these cases, and the investment prospects are therefore very low. So getting this funded is a real difficulty. However, if there is evidence that these existing drugs could be useful in the case of, for example, SynGAP1 missense, then a clinician can decide whether or not it's reasonable to use it off-label. And this has actually already been done. We can give as an example statins, which have been prescribed for SynGAP. And as far as I understand, in the complete absence of data for whether or not the statin could really be useful for SynGAP itself, it's more that statins have a particular effect on lipid metabolism, which was thought to be potentially useful. And it's been used in epilepsies before. Okay, so our project is called Identifying Targetable Defects in SynGAP missense variants. And the reason for this is that we are trying to make possible these small molecule screens, particularly with a drug library screen in mind, to identify those drugs that render SynGAP variant function more like the wild type. So what we're talking about here is an assay development project. We're trying to get to the stage where we can run a drug screen. We wanted to try to include the budgeting of the drug screen into the project, but without having the assays and without knowing what that entails, it was actually not possible to kind of define a budget where we could be sure we could go ahead with that. So this project is to get to that stage, to have those assays ready, and hopefully proceed with a drug screen in a future project. Okay, so we have three aims. The first aim is to monitor the localization, expression, and dynamics of SynGAP variants compared with the wild type. The second one is to try to understand the mechanisms of mislocalization, because we kind of know that there is some level of mislocalization, and to look out for aberrant gain of function, because if the protein is not in the right place, it may start to interact with proteins, or maybe because it's interacting with other proteins that we actually don't know about at this point. And by applying appropriate methods, we may be able to identify those, and that interaction may actually be a druggable target to follow up. But we have to identify those first. And the third aim is to apply structural methods, because these are still important, to understand the disruption of protein structure in SynGAP missense variants, with a view to possibly looking out for restoration based on the structural data. However, I have to say that the tools for protein structure and ligand docking and so on are not really ideal for identifying structural restoration approaches. So this is really a bit of a long shot, but we thought while we're looking at the protein structure, it's definitely something that we need to consider and take a look at. And in our project, which is really a kind of proof of concept for this assay development, we are looking at these three missense variants, so we don't have too wide a range to consider. Okay. We stuck. Okay, here's the next slide. So the first question is really, what are the functions of SynGAP1 that we can take a look at, and how can we measure them in a scalable way? Because we want to do these screens in the future. We want to measure, use hundreds or even thousands of different drugs. So we need to be able to assay hundreds or thousands of different samples in parallel. Okay, so first of all, SynGAP1 protein function. So SynGAP1 is a GAP. It's a GTPase activating protein. So what it does, it enables or it promotes the GTPase activity of the small GTPases of the RAS family, such as RAS itself, which is normally bound to a GTP, and it's in active form. And then after a GTPase action, this will form a GDP, and RAS bound to GDP is in inactive form. So while SynGAP is a GTPase activating protein, what it's doing is in fact inactivating the RAS. It's turning it from this form to this form. Okay, but this is just the GAP domain. There's a lot of other parts of the protein. And to go into what the rest of this protein does, we have to consider not simply the action of SynGAP on another protein, on RAS, but on the cellular context. So here we move over here. So SynGAP1 is a synaptic gap. It's primarily localized to excitatory synapses in the brain. In other places too, but we're not going into that now. So I'm drawing a synapse very schematically over here. So normally we have SynGAP tethered to the receptors near the membrane, synaptic membrane. RAS is at the membrane. And what SynGAP is normally doing is turning RAS into an inactive state. But when there is synaptic activity, there's a relocalization of the SynGAP away from the synapse, which enables RAS to have a chance to be activated, to act on its targets, such as the ERK pathway, which then in turn promotes the externalization of neurotransmitter receptors. An increase in neurotransmitter receptors at the membrane means that the synapse is now more sensitive to a neurotransmitter release. So there's increased connectivity, increased firing of the neurons and the synapses, and the promotion of network activity overall, which can then further promote the relocalization of SynGAP out of the synapse. So you can see there's a positive feedback loop going on here. So it's perhaps no particular surprise that if something goes wrong and there isn't enough SynGAP to reduce the activity of RAS, then we can head towards a an epileptic state or seizure activity. So if we think about brain function, for example, this can be measured in mouse models of SynGAP. We can start to look at the cognitive roles of SynGAP1 and activity and so on. So this is great for validation of drugs should we get to that state, but it's not ideal for screening. We're not going to be testing hundreds or thousands of drugs in mouse models, but we can consider this in cell-based systems. So we're going to stick with the cell-based systems here. So I want to highlight also that each of these steps leads on to the next step. So we should be able to read out if there's an impact on SynGAP1 localization, we expect to have impacts downstream. If the relocalization is faulty, then we should also have these downstream effects. So we can measure this at multiple levels. Maybe we should measure at multiple levels, but even one level will already tell us if something's wrong. Okay, so in our project, the aim one is to set up assays for localization, relocalization, expression stability, solubility, and the activity of SynGAP variants on signaling, such as ERK. Once we have these assays set up, it should become fairly straightforward to then ask, okay, so this is the wild type function now. Does the presence of the variant, which maybe has a disrupted function, does it actually prevent even the wild type from playing its normal role? This is the dominant and negative actions that we will look at. Okay, and aim one is this is mainly has been carried out so far by Lily Lee in the lab with help from Barbara and myself. Aim two is where we start to work with the neighboring proteomics facility of Otto Kauko and his colleagues. We will set up proximity interaction screens. So Lily is very familiar with proximity labeling methods already, and to put this into the proteomic context means that we can now, in an unbiased manner, evaluate all the proteins in the neurons and ask which one of these are interacting with SynGAP. Are there new proteins interacting with missense variants of SynGAP that are not normally interacting with the wild type SynGAP, because they may well be targets that we can follow up. Okay, and then, as I mentioned already, we have the SynGAP modeling team to predict the structural perturbations caused by the missense. So this is Alia Ali and Pekka Postila in Olli Pentikainen's group in our university. Okay, so before we go into the results that we have been obtaining, I want to show later on some preliminary results. That, please bear in mind they're preliminary. We haven't finished our project. The work is still going on, but we have some indications of where it's going and what's working. But before we go there, I want to a little bit take a look at the literature with you to explain why we are taking the approaches we are and why we're perhaps not taking another approach, at least not as a main path, let's say. So SynGAP is a gap. It means it causes inactivation of GTPases of the RAS family, such as RAP1. So it's possible to express the gap domain alone. Actually, that doesn't work for some reason, but the C2 and the gap together can be expressed. Just this fragment of SynGAP can be expressed in bacteria, purified to a high level, and put into a tube together with RAP1 in this case, and an optical assay system. So we're talking, sorry, I'm showing the structure here because we're talking just about this core domain of SynGAP. So none of these other parts are present in this assay. This is work that's been published by Mingzhi Zhang's lab. And you can see very nicely in this chemically defined purified system, we can easily measure, or they can easily measure, the activity of SynGAP C2 gap fragment on a substrate RAP1b. And if they look at a missense mutant in the gap domain, it turns out this residue is fairly important for the gap activity, because you see the green line here from this missense. It's much less active on RAP1b. And then they also looked at a couple of missense variants on the C2 domain. These have a lesser impact, but anyway, quite a measurable effect on the wild type function. So this is very clear. It's a very nice, simple readout. There are some issues to bear in mind, though. So this is it's completely cell-free. So it's lacking the cellular environment. So whatever it is the cell might do to this system is not measured here. preparing these proteins in a purified form is actually a little bit laborious. So if we would have a lot of missense variants to look at, it could be quite tough to go through them all. We are only considering this fragment alone, the core C2 gap domain, tandem domain, and ignoring the rest of the protein, which may modulate this function. And the expression conditions are artificial. I said these are expressed in bacteria. So what are the limitations that we want to avoid in our approach? So these kinds of methods actually require quite a lot of protein. So we cannot simply extract the protein from neurons or from brain. We have to use so-called expression systems, and bacteria are the easiest example, but there are plenty of others. And we actually already know, it's well known that some wild-type proteins from human or from other cells, they fail to express and fold properly at sufficient levels in these expression systems, because the proteins are not that easy to express in high levels. The cells actually have trouble doing that. So that doesn't mean there's anything wrong with those proteins. I'm talking now about wild-type proteins. So in fact, full-length SynGAP is one such example. You can't express full-length SynGAP in a native form in E. coli because it isn't soluble. But you can express this domain as was shown in the previous slide. So the failure to express is not actually an indication of a problem with the function of the protein. It could be a problem of the expression system, or the problem with trying to produce too much protein. So that's a complication if we want to use this system to understand missense variants. A second issue is that if we have activity, or let's say if we don't have activity with the isolated protein, is that the same as what's going to happen in intact cells? And here we have to think about chaperones. So chaperones are proteins in intact cells that promote folding. Human cells have them. E. coli also have them. They're not the same chaperones, in fact. So we may have a protein that is requiring human chaperones to fold properly, or let's say a missense variant may still fold and be functional in a human cell or in a neuron, and if we isolate it from bacteria, it actually may not work. So there are some of these unknowns that are going to confuse us a little bit. So though it's a really valuable method for targeted screens, let's say we see that the missense is not working well, and perhaps we want to try and find a molecule that improves that, we're taking a more phenotypic approach. We're trying to understand, in the context of a neuron, is a missense variant working well, or is there something going on? So that's why we're not going for this. But having said that, there are some specific questions that we may need to address using recombinant protein, and it is quite a lot of work, like I said, but we're very lucky that these questions have come up for us at a time when our university has just set up a protein core for which they can provide services to us researchers, basically to generate proteins for us. And of course, we need to pay for that, but in this early phase, they want to do trial runs to demonstrate their services, and they're not charging us for that. So this is perfect. We can get them to produce some SynGAP protein for us, or the C2 gap domain in this case, and Pedro at the Tuco protein core has been doing just that. So that's really good. So we've been able to make use of this opportunity to help us in the SynGAP project. Okay, so one step up from recombinant protein is to look at SynGAP in a model cell line. There are various ones that could be used. Many of them are human cells, or even better, not just a mammalian cell, but a human cell line. They have their own gap, they have their own MAP kinases, and so on. And the HEC293 cells, we like them. They're really easy to use. We use them a lot in the lab. It's very easy to express proteins on them. And then we can do all kinds of measurements. So in fact, there's a very nice study, a very comprehensive study from Kurt Haas's lab that has done just this in HEC293 cells with 57 different SynGAP1 variants. It's pretty amazing work. And he has, or his lab has shown, amongst other things, how the stability of the protein looks in the case of all these variants in comparison with the wild type shown in black here, and what is the activity of various pathways, including, let's look at the ERK pathway here, of the wild type compared with the different variants. Now, not only black is a wild type, but there's a few groups here. And so we have to look at the blue columns which are missense variants associated with disease. And the green ones are actually missense variants not associated with disease. So we can see, for example, in the stability plot, there's actually not a huge difference with a couple of exceptions. So actually all of these proteins, all of these variants are able to express. So it's not like the nonsense and nonsense-mediated decay at all. There's actually pretty good expression of most of them. But the disease-associated variants, well, some of them, they have a slightly reduced expression, and some of them have a slightly increased expression. So we can't say that, okay, there's simply not enough protein around, at least not in most cases. If we look at the ERK pathway, so we can see that this effect is actually a little bit on the small side, but certainly the wild types on the left-hand side, meaning it's more active than most of the others, but many of them are not really very different from the wild type. And those which are most affected here include some of these missense variants that are not associated with the disease. So it's a very neat system. There's a lot of data here to look at. There are disadvantages. It's not a neuronal system once again. We don't have the neuronal compartments. We can't ask about SynGAP at synapse because there are no synapses. This is actually a kidney-derived cell. It's a very convenient system, but not a neuron. And this is an overexpression approach. That is that these are normal cells, and there are methods used to overexpress SynGAP because there's no SynGAP in these cells under normal conditions. So if we go to these cells and we transfect them with SynGAP protein, so you can see the cells here in blue in the microscope, and we can see the SynGAP expression in magenta. We see the higher levels, at least here. And what we notice is there are pretty large aggregates. Now, to be fair, if we look at neurons and we stain for SynGAP, we do see puncta. It's not like we have a smooth distribution. We see SynGAP in puncta, but some of these puncta or blobs are really huge in the case of the 293 cells. They're almost halfway across a cell. So these are perhaps large aggregates that are perhaps not what we would normally see in a neuron. Now, we can control the size of these aggregates by adjusting the conditions, by maybe trying to reduce the level of expression, but it's hard to know what is not overexpression in these cells because they normally don't express any SynGAP. So this introduces a little bit of uncertainty here. And again, it's a reason why we don't really want to use these cells in this analysis of missense variants. We actually want to take a look at the neuronal systems, even though the 293s are a great method, a great system to work with. Okay, and so we were largely inspired by this early work from Gavin Rumbau, who showed that if you take neurons and culture them from mice, or if you take neurons from, these are wild type mice, and these are mice embryos, mouse embryos that have a complete knockout of SynGAP, you can very easily see a big difference in the ERK activity. So what we're looking for in our case is a high throughput approach. It starts to get complicated if we have to have different mouse lines, especially the full knockout is actually embryonic lethal. But what we can do is we can, in this case, we can take rat neurons, we can put them into our high throughput microscope system, automated system. We can include an ERK reporter, shown in blue here. When we stimulate synaptic activity in the cells at this point, time 0, then we see an increase in ERK activity. It's easily measured. And we can introduce reagents, in this case an AAV, to knock down SynGAP, it's an RNAi, and we can see an increase in ERK activity. So this all looks good, and it's, as I said, it's automated. So we have the authentic neuronal environment. We're talking about the endogenous protein. This is real SynGAP in a cell that normally expresses SynGAP. Neuronal compartments exist. There are synapses here. We can go in if we want to and introduce missense SynGAP. Nevertheless, there's still some issues here. So, for example, this SynGAP, yes, it's SynGAP, but it's not human SynGAP. We have the wild type there. We don't have all the missense. Having rats that already express missense SynGAP instead of the wild type would be problematic and difficult to achieve. But we have to bring them in on top of the endogenous SynGAP. And then these are actually whole cell measurements, both in Gavin's paper and in our study. So the synapses are there, but what we're actually seeing is that ERK activity all over the whole cell is being increased when we reduce the levels of SynGAP. So we're not getting a synapse-specific readout of SynGAP function here. We can also go to human cells, and I'm taking this example from Gavin Rumbaugh, who kindly provided us with his cell lines so we can produce human neurons in our lab as well. And here's one of the readouts from his paper on this. So you can see in this particular graph that there's a difference between the red and the blue lines. So there's an increase in synaptic activity in the knockout lines compared, or the knockout neurons compared with the wild type neurons. But the effects are a little bit modest, I would say. And it's only through combining two different lines of wild types, two different lines of knockouts, that statistical significance is achieved with this particular measure at this particular time point. So there's other data, some of it's a little bit clearer, some of it's a lot clearer, in fact. But depending on what you measure, some of the effects here are a little bit on the small side. But it is a real human neuron system. It's a human SynGAP that's being expressed here. There are neural compartments once again. But we thought that this perhaps may not be the best system for screening. It may be good for validation and for testing particular points, but and that's what we plan to do. We will try and use human neurons for validation, but not for screens in this case, for the missense variants. I want to highlight the mouse models. Obviously, we're not talking about mouse models for screening thousands of drugs, but there are some very useful models have now have been developed by Rick Hugen's lab and Gavin Rumbaugh's lab. In these cases, they are expressing these human variants. In this case, there's a frame shift and a point mutation leading to a truncation. And these will allow and do allow behavioral cognitive measures, which is what we care about in the end, after all, and investigation of brain circuits and so on. So for at the validation stage, should we come up with some molecule, such models could be very useful. But what I want to highlight here as well is that these studies have already shown that it's the SynGAP alpha isoform that appears to be particularly important for restoring higher functions, such as these cognitive measures. And that's important because it's the alpha isoform that we're studying in our research as well. Okay, so we're talking about setting up screening assays. It means automation. It means that we need to have the SynGAP missense variants expressed in all or most of the cells. We need efficient gene transfer. And the best way to do this is to, it seems to us, is to use the adeno-associated virus system. For gene transfer, it has low toxicity, prolonged expression, it's very efficient, very easy to work with. Everything's good, except the genome size is very small. This is a problem that we had to deal with. The first thing we had to deal with. So the typical off-the-shelf AAV vector that you can get hold of is not even big enough to put in full-length SynGAP. So then we can't study the wild type compared with the missense variants because we cannot put in SynGAP into the AAV. And that's before we think about adding a fluorescent tag, because we want to look at the living cells, to look at dynamics and so on. So the first thing we had to wrestle with was how to fit SynGAP into an AAV. We can fit it into the AAV plasmid, but AAV particles will not form unless the cassette between the two ITRs, inverted terminal repeats, is small enough. So the whole thing can be about 5,000 bases, but not more. So to cut a long story short, we try to optimize all these, all these non-coding regions so that to enable us to introduce a large transgene, which is SynGAP in our case, and we were able to save about 1,000 bases of space compared with the typical commonly used AAV vector, while still retaining most of the expression levels, or let's say about half of the expression levels. Okay, and then that is good, but we still didn't have room for the fluorescent protein, which is another 750 base pairs that we have to introduce. So we can put in SynGAP, but we want to see where it is. So we want some kind of fluorescent marker of it. So for this, we turn to fluorescence complementation. And in this case, we have made use of the observation that you can cut a fluorescent protein, which looks something like this, a kind of barrel shape, into two parts. And in the best case, we can have a single strand here of about 15 amino acids, and the other 250, 220 amino acids or so over here. And if they interact together with one another, then we recover fluorescence. So all we need is to have a SynGAP, a very large SynGAP protein with this very short tag. And if this rest of the fluorescent protein, which is introduced with another virus, if it's present in the cell, then we will get fluorescence where SynGAP is. In order to ensure this interaction takes place, we actually need to have an affinity tag here. So we actually have to have both this orange part and the red part. But anyway, it saves us about another 650 base pairs of space. And then finally, it means that we are able to introduce SynGAP with not a fluorescent tag, but a fluorogenic tag into neurons with AAV. And if we also introduce the rest of the fluorescent protein with this affinity construct, then we can generate fluorescent detectable SynGAP. So this is an immunoblock to show that the box here is in the way, but this is just the uninfected cells with the endogenous SynGAP. Rat neurons normally express SynGAP, and it's about 130 kilodaltons. If we then introduce the AAV system to express the SynGAP with a fluorogenic tag, then we have this larger form of SynGAP which has a fluorogenic tag covalently attached. And we also do see an increase over here. So this formation of the fluorescent form isn't 100% efficient, unfortunately. And this may cause some complications in some cases, but it's anyway useful enough that we can carry out some assays. And these are the very first kinds of images we got with the very initial constructs. They are not the fully optimized and corrected SynGAP sequences. So I'm putting wild type here in quotations, missense 1 and missense 2. So these are anyway showing that the constructs allow us to visualize in living cells a localization that looks quite normal for SynGAP along processes, a little bit in the cell body, whereas the missense variants seem to be somewhat different, let's say. Although there are puncta here, there are perhaps just fewer of them. Okay, so now we know that the system basically works, and we can put in all this optimization, and we need to make sure that the SynGAP sequences we are using are absolutely perfectly correct. Fortunately, there are new full plasmid sequencing services available nowadays. We don't need to go for Sanger sequencing, and we can check what we have is really correct. And we've been able to produce and validate 6 constructs now and use them. This is the SynGAP1 alpha 1 wild type, 3 missense variants that are part of our project, a nonsense variant as a comparison. I want to highlight that normally if one would have this nonsense mutation in a human cell, it would be degraded, like I mentioned, by nonsense-mediated decay. But because we're just expressing the coding sequence, we don't have the extra untranslated regions in our AAV. We don't have the nonsense-mediated decay pathway kicking in here. So what we will see with this variant is an expression of SynGAP as it would look if the SynGAP1 was truncated without the nonsense-mediated decay. Okay, and we also have the SynGAP1 alpha 2 wild type as well. We've designed this in a modular way, so it's very easy to, for example, put in these mutations into the 1 alpha 2. We can switch the split fluorescence protein tags and have alternate affinity tags for the double labeling approaches that we will need for the dominant negative investigations. We want to have alternative complementation strategies to try to achieve higher efficiency of labeling, which could be important in some cases. We are working on the proximity labeling tags that we need for the proximity proteomics. in AIM2, and we have a variety of other variants that we're working on. So we have quite a number of other constructs ongoing, which have not yet reached the stage of full validation, but they'll soon be available to us, and we'll be able to use them in our project. Okay, but I want to give you an example of what it looks like in terms of the localization, which we're measuring together with the longitudinal stability, which I will explain. So just to remind what it is that we're trying to do here, we're trying to look at the missense SynGAP and understand what is wrong. In this case, identify the localization of the missense SynGAP in comparison with the wild type. And it's very hard to kind of manually look at these images and say, okay, this looks normal, this doesn't look normal. We can kind of say that, but if we start to now introduce drugs and maybe there's a shift from one state to another, we need to really numerically quantify that. And that's what I want to show. And once we have this numerical quantification, then we can start to go for the rectification attempts. And yes, what we're trying to do is to have some kind of system which allows us to screen up to 4,000 drugs. And we have to bear in mind that some readouts may just be too noisy, and it will not really be possible that other readouts hopefully will be less noisy and robust enough to do that. So this is what we need to do. Can we find a way to make the readouts sufficiently robust for this? So we are using complex cells, which are not that easy to work with, and certainly not as easy as a 293 cells or cell-free assays. So 10,000 is actually not very many samples in a screen for a cell-free assay, but for complex cell models, it's actually quite a lot. So we need a simple assay, fully automated, high throughput, we need to demonstrate high reproducibility of the readout. Ideally, and it's commonly done with a robust screen, you just do the screen once and look at the hits and work with them. We may have to do the screen twice or maybe 3 times, and just make sure we're not generating, the worst case, it can generate a lot of false positives. That is, find compounds or drugs which we think are useful, and then when we go to do all the validations, we find out actually, no, that was wrong. We had some variation in our screen which made us, which fooled us. So we really don't want a lot of false positives because we'll spend the rest of our lives trying to figure out or find out that they're not useful. Okay, so as an example of the development that we're doing here, I want to show you one of the tests we ran. So we had 7 constructs, that is 1 wild type, 3 missense variants, a nonsense variant, and some additional controls. And then we were testing out 4 different detection methods using these split proteins, and we had 6 different replicates in each case. So all this together leads to 168 different samples. We put them in a 384 well plate, which is a plate about this size, which has little sample wells in them, about 3 millimeters across each. And for each sample, we took microscopy imaging fields, 9 of them, neighboring fields with 9 to 13 different Z positions, so we can get a 3-dimensional representation of about 10 to 30 different cells. And what we wanted to know was this measurement that we're making reproducible, so that if we measure it today and we measure it tomorrow, would we get the same result? It's no good if we get one result one day, but if we measure it another day, we get a different result. That's something that we'd need to know. So that's why, in part, why we're working with living cells here. So the cells are still alive tomorrow, still alive the next day. We're using AAV, so we can actually see expression for weeks in the cells, which is very convenient. So we can do this. So we have measured actually 5 time points in our original data set to check whether or not the readout is the same day after day. So this particular data set comes to about 100,000 image frames. So the image analysis, obviously we're not doing this manually, drawing circles or counting spots. We need to find a way to automate this. And what we noticed, and what you might have seen in the earlier slide, these little spots of SynGAP, they're really quite diverse. You can see the bright ones. There are sometimes some really bright, bigger ones, and then there's some very dim ones. And that means it's actually quite difficult for conventional segmentation approaches used to detect spots to find the dimmest ones and the brighter ones and all various different kinds. And that's why we went for training a deep learning model. I don't go into this unless someone's interested to hear more about it. But we were able to develop a deep learning model that could be used in imaging software that's commonly used called Fiji or ImageJ. And our model in the end that we were able to train to identify the spots were in a sense too good. So on the one hand, everything that it detected, we took a look at some examples, and it really was the puncture that it was detecting. We couldn't say at any point that, okay, it's detecting something that definitely isn't a puncture, but it was detecting even the really tiniest ones that we wouldn't normally include in our analysis. So I think that's okay, because we can just gather all the data and we can say, okay, now we don't want the smallest ones because there's no way they can be synapses. It's just way too small. So we need to add filters to our data during the downstream analysis. So we were able to do this in the identification of the puncta. We want to separate those puncta that are in the cell bodies of the neurons from the ones which are along the dendrites and so on, because we're interested in synapses, and we can't be sure that's what's going on in the cell body, but in the along the dendrites, we can be more sure that we're dealing with synaptic puncta. And in the end, from this data set that I described, we had a total of about 35 features per puncta, leading to about 1 billion values measured overall. And this is quite a lot, obviously. So what I can say is the segmentation was actually pretty efficient and very fast. The model was very good and worked well, but the feature collection, collecting all this data, was really quite slow. And this could become a bottleneck if we want to measure from hundreds or thousands of drugs. So this is definitely a concern, but we're quite fortunate in Finland that we have this organization whose job is in part to provide researchers with free access to supercomputers. So we can, without charge, use supercomputers for running this kind of, getting through these kind of bottlenecks. And not only that, but the third fastest supercomputer on the planet, according to a measure last year, is in Finland, looked after by these guys. It's owned by a European consortium of 10 countries, but we also have access to this one. And what's more, these guys, Lakshman and Ate, have recognized that image processing is a growing area requiring high performance computing, as they call it. And so they have put aside some resources, that is themselves, to help us bring our image quantification scripts into a form that's applicable to the supercomputer, because we can't simply put those scripts onto the supercomputer. They have to be converted in a particular way, and that's something that is something these guys are very good at, and they're helping us do that basically for free from our side. So it's another resource we're bringing into the SynGAP project, luckily. Okay, so I just want to give you an example of the kind of data we end up with from these billion data points. So we can just pick one feature. Let's say we want to look at the mean intensity of all these puncta we've detected. We can make an average for every sample. We had 168 different samples. We're just considering those with one detection method. And now we can compare the 6 replicates of the wild type with the different missense variants, and it's pretty obvious there's a difference, and that's good. So we can see that even measuring the mean intensity, there's a clear difference, which is what we need. But actually for a drug screen, this level of variability is actually even a little bit high. We want to have something more robust, but we have all this other data. So the question is, what do we do with all this data? How do we handle all these features, and how do we do filtering to get the most robust possible readout? That is, we want to get these error bars as small as possible. We want it to be as clear as possible what the difference is between a wild type and a missense variant. And ultimately, is the data so reproducible that just by looking at those numbers, can we see, okay, that's this form of missense, or that's a truncation mutant, or that's the wild type? Because once we get to that stage, then we can say, does this or that or another compound bring the results towards the wild type state from the original missense state? So what we want to do is make use of all the relevant data. We can talk about what is relevant and what is not relevant among the features, but this is what it looks at as a kind of summary, looks like as a summary. So what we did here was ask the computer to look at all the data and represent those features for all the data without telling it which ones are the wild types, which ones are the truncation mutants, which ones are the missense variants. Just position every sample according to the properties of the puncta. on a 2D space, and this is called a UMAP. And so what we did when we looked at the result, we found that, in fact, all the truncation data from the truncation variants are all over here, all the data from the missense variants are over here, and all the wild-type ones are grouped over here, and they're not overlapping at all. So this is good. This is just the first step. It gets better. But the other thing we wanted to do, I mentioned we were measuring repeatedly over multiple time points to check the stability. So what we want to know is, are these data points very noisy and going from one place to another over time? So if we simply draw a line between the points, so here we have the first data point, second data point, third data point, fourth data point for one particular sample, one particular well in that 3 or 4 well plate, one particular field of neurons. And we can see that by and large, those samples are kind of keeping within their little space, which means they're staying very similar over time. There are a couple of cases where there's a bit of a jump here in this sample and a couple of missense variants switching from one group to another. So there seems to be 2 groups here, but by and large, they're really staying in their space over time. And this is important for drug screening because we know that if we would test a large number of constructs, it will take a long time. And we do not want our assay to suddenly show something different later on compared with how it was earlier on. So we know we can put our plates in the machine for 5 days and everything keeps in the same space. That's actually very important. And what about filtering? So we didn't filter anything. We just allowed use of all these little tiny spots as well, which are not really what we probably want to look at. So there's a question, where are the cutoffs that we should use? So this is the result of putting in a cutoff, and the cutoff was based on a kind of experimental evidence. It wasn't us that, well, we chose it, but we chose it based on empirical evidence. So that's important. This is not any kind of supervised machine learning. We're not overfitting here. What we're doing is we're saying, okay, we actually realize the small spots are not what we want. We only look at medium-sized spots in the data. And we ask the computer to, again, sort out all the samples according to the properties of those spots, again, without telling the computer what are these different variants. And we can see we get really good separation between the wild types, the truncation mutant, and the missense mutants. There's definitely overlap here between the missense mutants, which is kind of interesting. I mean, they could have been very different, but it turns out that they're actually very similar, even though there are different mutations in. It seems like this one, I think, is the LR mutant. It's a bit off to the side, but I think this tells us already that we're kind of ready for a drug screen with this kind of readout, because there's such good separation between the replicants. Okay, so what else can we do? So it's not enough for the SynGAP to be at the synapse. It actually has to disperse in response to activity. And that's why we're using living cells, because we can ask that question. So really beautiful work from Hugenel's lab showed this dispersion response. So the question is, can we actually see this in our system in an automated fashion? And sorry, I've got the time scale labeled wrong here, but under baseline conditions, we have this kind of distribution of SynGAP. This is the wild type, and we can induce a synaptic activity. And what we see here, the movie's not working. It's supposed to be a movie here. Sorry about that. But oh, yeah, is it running? Okay, now it's running. So you can see these little spots kind of get a bit fainter, and the neighboring regions get a bit brighter. So this is quantified here. So we can see at the end of the time course, there's not really very much difference between where the puncture was and the neighboring region. So this is what we expect. It means that, okay, that we can, in an automated fashion, do automated pipetting on our samples and read this out from all of our samples. And we can do this in high throughput. And this is some data. It's just a very preliminary data. It's definitely a little bit noisy, much noisier than we would like. And there are things that we can optimize, but we can clearly see with the wild type, there's a clear dispersion, there's a response. The number of puncture is reducing over time. And this is where we realize, actually, it's the medium puncture that we want to look at, because if we look at the small puncture, they're not really responding. But in terms of missense, let's say a preliminary conclusion, we have to be careful here. We need to do more work to be sure, but it looks very much like all those variants, they're also responding. They're also capable of localizing, capable of responding. Maybe the ALR is a bit defective in the response. Maybe this is a little bit steep, but we really need to do more experiments to be sure. The small puncture, that's kind of interesting. They seem to have bigger responses in the variants than in the wild type. But let's see. So whether or not this is going to be a screenable readout, I don't know, but I wanted to highlight one more thing. It's a bit cumbersome, as in the last example, to actually have to pipette on a stimulus in a high throughput manner to monitor the response. So in the first case, we were simply doing imaging. In the second case, we were doing imaging during a pipetting using robotics on the imager. It's doable, but it's not so scalable. So it'd be much neater if we could use optogenetics to cause the translocation. So the Rick Hugen's lab has demonstrated what is the mechanism behind the translocation, which helps us to develop an optogenetic method. And here's the outcome of this in some preliminary experiments, some setup experiments. So the magenta here, you can see the spots of SynGAP at the beginning of the experiment. You can see the little spots along the dendrite. The cyan is the location of the dendrite. And in fact, I've already given away the result because it's actually the final location of SynGAP. So in this experiment, we're simply using light to switch on the diffusion or the dispersion of the mechanism causing dispersion of SynGAP. And we can see in this movie here, hopefully you can see those magenta spots disappear pretty quickly. Okay, so one last slide. This was from our SynGAP modeling team. Pekka Postila provided this slide to me today. So what they've been looking at is using molecular dynamics simulations. Do you remember I was saying that when we look at the predictions of SynGAP structure, there are some regions we can predict, some regions we can't predict, but among those regions we can predict, and even the crystallographic data, it's very static. It's just a kind of frozen condition of the protein, and in reality, the protein has flexibility. It's moving around. It has to move around to do what it's supposed to do, to have its functions, to be a gap, for example. And so what the modeling team did was to use molecular dynamics simulations software called AMBER to consider how does the wild type move compared with the missense variants that we're looking at. And they can see quite rapidly that within a few nanoseconds, there are really significant conformational effects of putting in these missense variants, and that perhaps could be predicted already because these are quite large changes at critical regions in the structure. Nevertheless, these are actually quite localized deformations of the structure. And what's seen, because the whole C2 gap domain, a pH C2 gap domain is modeled here, there's no large scale unfolding. So this is kind of important. It's not that these variants are causing the whole protein to collapse because, or even the C2 domain to collapse, but it's just kind of causing a local disruption, and the rest of the domain seems to be able to cope with it. Now, it doesn't mean that the domain is functional. That's another question that has to be addressed, but at least what we know is that it's not driving misfolding of the protein. Okay, now with that, I would like to acknowledge the people who have been behind this work. So for the experimental work, it was mainly carried out by Lily with help from Barbara and myself. We've just recruited Pierre to the lab. He's going to join us next week. Now we have all these additional tools for SynGAP that I described in AAV. We have a lot of work to do now, and we really need more hands on deck, and Pierre's going to help us with that in the future. And the SynGAP modeling team, that is Alia Ali and Pekka Postila in Olli Pentinkainen's lab. I forgot to say they also have a new SynGAP, a new SynGAP project funded by SRF to look not just at those 3 missense variants, but to do predictions on a much wider range of SynGAP missense variants and develop a web portal that will help clinicians make decisions as to whether variants are potentially pathogenic or not. And the proteomics will be done with Otto Kauko and his team. I mentioned Lakshman and Atte helping us with supercomputing, and Jukka and Gani help us with some additional computational issues. And Pedro I mentioned with the Tukul protein core. And this work wouldn't have been possible without SynGAP Research Fund and Leon and Friends funding. So thank you very much for listening. I'd like to take some questions if you have any.
Yeah, that was great. Thank you so much. That was really good. There's one question in the Q&A real quick that can you see it or I can read it. Says, very interesting talk. In your AAV vector, did you use full-length SynGAP1?
Yes.
And what promoter did you use for the SynGAP1 expression construct?
Right, that's a good question. Yes, so yes, we did use full-length SynGAP1. The downside is the space is definitely constrained, so we're not able to introduce untranslated regions, which are potentially important to help regulate the localization. For example, they could be local translation at the synapse. So we unfortunately aren't able to introduce some of these additional aspects with AAV. Okay, and then the expression constructs that we're using in the end, we decided to go for EF1S. So there's quite a commonly used promoter for a housekeeping gene, elongation factor 1. It's about 1.5 kilobases normally, but there's an extra short version that's been used for Cas9, which is huge as well, which is only about 250 base pairs long. The downside is that it's not particularly neuron specific, but we are using neuron specific promoters for the complementation part, so that we only get fluorescence complementation in neurons because we have a synapsin promoter, for example, in the complementation AAV. Hopefully that answers the question.
If anybody else has questions too, yeah, go ahead. Or if JR, I know she's here, or if anybody wants me to promote you to panelist, I can definitely do that too, and you can answer or ask yourself. If you want to go ahead, JR, though.
Yeah, hi. Thanks, Lauren. Hi, Dr. Courtney. Thank you so much for that amazing talk. And Lauren, please, interrupt me and have anybody. I'd love it for you to continue with the managing the questions, but I just wanted to
For sure.
Say a few things. First off, wow, thank you for such amazing work. And I really loved how you went into really, really good detail on different types of assays and why, why some are appropriate and why some are sufficient or not sufficient. That was really, really helpful because I think a lot of us as parents, I'm a parent, a lot of us are wondering like, why isn't there an assay? Why can't we do, what's going on? Let's just look at this. Why are we having to raise money? Why are we having to give so many cell lines? And I think you really gave a lot of detail around what the tough issues are and why it's tough to figure this stuff out. Your data where you were showing the colored spots coalesce, if you could kind of maybe go back to that a little bit and tell us, just kind of remind us really simply what's going on here. So I think you're looking at, I understand the wild types. I understand the RX is the nonsense, right?
Yeah.
Is that nonsense? Is that one that's not going to have nonsense mediated decay? Is that right?
Normally it would have nonsense mediated decay if it was in the normal genomic context. But in our case, because we don't have these untranslated regions and splicing going on, then it is being expressed kind of aberrantly, if you like.
Right. And so the wild types are being expressed the same way as the missense, right? That all these versions are being expressed the same way. Is that right?
They're all being delivered to the same neuron culture. I mean, there's a neuron culture which we put into a multi-well plate, and we have some wells where we put in the wild type, the virus encoding the wild type. And in fact, we have two different, if you're wondering what this purple spot and pink spot is, one of them is the corrected wild type. This wild type E is correct, but there was a silent mutation which was just in the DNA that we were working with. And although the silent mutation means that the protein should be correct, we just thought maybe it's better to have the completely correct sequence. So we went in
Right.
and
Well,
corrected
you
that, but
never,
we had both.
yeah, so you never know. So in
Yeah.
fact, we've even seen some silent I've seen one silent mutation in SynGAP be categorized as pathogenic, and I don't understand why. Like
Okay.
I understand
Yeah.
possible reasons why, right? I
Yes.
understand
Yeah,
possible reasons why. I don't know why in particular that one was, or whose decision
Yeah.
it was, or what they were seeing that made them think that.
Yeah.
But yeah, okay. So I guess there isn't a way with this system to get to the nonsense mediated decay.
We haven't really thought about that. So how would we get to it? You mean, I mean, I think
Well, you'd have to have
Okay.
your cells, you'd have to have your cells start with it. I think.
Yeah.
Well, I guess not, because you have to
I know
have
what you
your,
mean. Yeah, I hadn't thought about that at all. So yeah, I mean, basically we're working within the constraints of the AAV system. So I think it's probably going to be tough if we want the full length SynGAP and we want to have those introns that are participating in the nonsense mediated decay mechanism. It just doesn't fit. So we could work with fragments if we want to specifically look at that. It depends what the question is. I mean, is the idea to ask whether there's a way to specifically express the nonsense mediated decay of a SynGAP variant? Or I think suppressing nonsense mediated decay overall probably isn't a good idea, right? Because it's a kind of safety mechanism.
right, I guess, okay, I have an idea. I think it would be expressing the wild type and co-expressing an untagged nonsense. I think that's what it would be, just to see if it changes the wild
Right.
type. Yeah.
With the nonsense mediated decay in place.
Yeah.
Yeah. Yeah, I think in principle, we could go in that direction with lentivirus. The problem with lentivirus, I mean, lentivirus has a lot more space and it works, and we use it for the human neurons, for example, all the time. But the problem with lentivirus is it has a different safety category. We have to work with it in a special lab, and with the human neurons, it goes through all these medium changes, and we start with a big plate of undifferentiated cells, and we end up with differentiated neurons that are kind of validated to be lenti-free. We can't really do that in a 3 or 4 well plate.
Yeah. I don't mean, I didn't mean for your, I meant just to do it once, not to do it as to do it as a, in a test.
Yeah. Okay. So that's definitely something that I think that could be done. We just haven't thought about it much, but it's potentially doable. So if you have a specific, do you have a specific nonsense in mind, or this?
No, not a specific. No, not a specific one. I was just wondering how that spot would change and how that spot might change the wild type. Because we're thinking, like, does that, the whole idea of the protein truncating variants not being dominant negatives is that they're not there.
Yes.
But in this system, they are there. So I was wondering if this is looking like some kind of dominant negative that doesn't normally happen in wild type. And maybe it doesn't matter. Maybe it doesn't matter if it's just, if the spots don't overlap with the missense, maybe it just doesn't matter. I just was thinking for certain, if you're going to test a whole lot of missense, you might want to know if any of them look like dominant negative, is what I was thinking. Sure, or if any of them look like, haploinsufficiency. And so those are the kind of the two, right? Those are kind of the two things you want to know. Do they look like do they look wild type? Do they look like dominant negative? Do they look like haploinsufficiency? Do they look like hypomorphs? Do they look like neomorphs? Right. And to see that, you're going to see spots in different areas.
Yes, that's correct. Yeah. So in a sense, except we haven't made the constructs in that way, but we kind of can almost immediately do what you're suggesting, which is basically to, let's say we express the wild type and we put in an unlabeled missense. So we don't even see it, but we know we put it there. And we can ask whether the wild type spots are kind of drifting away.
Right.
That would indicate a dominant negative effect. What we tried to do is to go for a double labeling approach. So we would see both at the same time.
Yeah.
Which is a bit more challenging. And so we were a bit slower in getting there. It's on its way. But yes, we could go for this kind of part labeled, part unlabeled approach. Yeah, that's a good idea.
Well, since your spots are so tight, you can.
Yeah. Definitely.
Okay. What are you most excited about here? Like as far as what do you like, are you most excited about a particular assay as being, you know, because as you said, you're trying all these assays.
Yeah.
You don't need all the assays to work. You need some, you know, you need one
Yeah.
or a couple to be really robust assays.
Yeah, I kind of like the way this came out. It was, I mean, I didn't know from the beginning how easy or difficult it would be to do the segmentation and to run the analysis. I think I was very happy with the deep learning model to identify the puncta. And I was also very happy to see this kind of come out without kind of having to resort to any supervised learning, because I'm quite worried that, I mean, I also dabbled with supervised learning methods, and you can see it just really easily overfits the data,
Right.
because it already knows, you know, which one's which.
Yeah.
And that's not what we want to do. We want the properties of the proteins to tell us what's going on.
Yeah.
So I'm really happy with the separation we have already. There's still some tricks we could play to try and improve it. But maybe we're already there and we can already make plans for the drug screens as it is.
Yeah.
I like the optogenetic tool for inducing dispersion. I think that's going to be just so much easier than having to handle the fluidics in a high throughput manner.
And so that, can you remind us that happens over a very fast time period, right?
I had an incorrect time scale. So actually, I don't really remember the time period. I think this is probably minutes, not seconds. Sorry about that.
Right, but still, that's very fast as far as like.
It is very fast.
Yeah.
Yeah. If we look at Rick Hugnir's data, I stole that for this one. Sorry, I'm blocking the screen for myself at least. So we can see in his experiments, which are endpoint measurements, I think they're fixing the cells and they're taking a look. They also see a very fast effect. So I don't know if that first time point is 5 minutes. Here it's 10 minutes over here, but it's a really fast effect. It's nice to have the live cell readout. I think most labs are not using the live cell readout because, of course, it's more convenient to fix samples and go when you can to the microscope and do more detailed studies. But what we're always trying to do is seeing what is it we can see with live cell, because the dynamics often tells us a lot more than just
yeah. So in the live cell dispersion, is that something you repeat, or is that something you do at once with the optical?
Right. So we've done this kind of experiment before, not with SynGAP, and it was really convenient that we could repeat it, because it's a bit like the electrophysiology studies. Quite often they just depolarize cells multiple times under different conditions and build up a whole lot of data, almost automatically in very little time. So ideally, we would be able to repeat this multiple times on the same sample and build up a kind of average and get much more precise information. Unfortunately, what we're seeing is that the reversibility here is pretty slow. So I don't know, I can't tell you how slow it is. And it may be if we have a lot of samples, that's fine. We just need to do this experiment on each sample, and then we can come back when we finished and do it all over again and do it all over again. And we would be able to get this kind of repeated measure that would give us more confidence and less noise in our measurements, which would make it more of a screening assay. It would take time, but it would still be doable. So all I can say right now is that it certainly doesn't reverse in a matter of a few minutes, which is kind of interesting.
Yeah, even with wild type.
Absolutely. Yeah, I didn't expect that.
Yeah.
But we are providing quite a strong input, as was Rick Hugnir's lab. So same idea here.
Have you thought about? Yeah, have you thought about doing less of an input and seeing what happens?
Sure. Yeah. I mean, that's quite easy with the optogenetic method. It's very easy. You can, set up all kinds of different stimulus levels. What seems to happen is you just get less of a response. But we need to do a bit more work. We'll see.
And then I had a question about the very small puncta that you were realizing had nothing to do with the synapses. Potentially they're in the cell body or in the, another part that's not the synapse. Is that right? Yeah,
but it could also be noise. I mean, we're a bit constrained by the amount of light we can put into our samples because they're living samples. Light is phototoxic, so we have to be a little bit careful. So we can't, that's another reason why people like to fix cells and they go to their confocal microscopes, and they can really use almost any laser power and they get away with it. But in our case, the cells will be dead after that, so we wouldn't be able to do it. So we have noise, which will give us little spots. So that is part of it. Yeah, there's also, I mean, if we look, let's go back to this dynamic measure here. The data is a bit noisy, but I think you can see in our data, and you can probably see, I think, well, it's kind of hard here, but in the Iraqi paper of 2015, they also have some data, I think, kind of indicating that the localization in the dendrites isn't necessarily totally smooth. It's a little bit punctate there as well. So perhaps you can see it here. What I've done here is it's the same data, but I've applied a deconvolution algorithm. This is the raw data. So perhaps you can see these tiny spots in the shaft, the dendritic shaft.
Yeah, it's like looking at the night sky. It's like dim stars, right? That's what it looks like.
Yeah. The other thing I didn't get to, maybe I can show it here, is that if I can just manually move along here, then you can see at the end, all the puncta that were here have disappeared, most of them, but these have stayed very strong. So we also have larger puncta of SynGAP that are not going anywhere with the stimulation. And I don't think it's because the optogenetics isn't in this cell. I think there's just kind of larger puncta. What they are, I don't really know. I mean, there's always a risk that we have a bit of overexpression in some cells and we have some artificial aggregates, or perhaps it's just a way. There is also SynGAP in the soma, in the cell body, which can form these fairly large puncta. I'm not sure if they're somatic synapses. I sort of doubt it. But so there are other structures here as well. Sorry, I interrupted.
Oh, no, I mean, the reason I was asking this at the beginning, I think you're probably gonna, I think I know the answer now, but I was gonna ask if you were gonna take that data of the smaller stuff and somehow separately try to analyze that for the cell bodies. But it sounds like if it's mostly noise, that's not gonna, you know.
We did try, yeah, absolutely.
Analyzing, oh, you did try, okay, yeah.
Yeah, we tried different size ranges.
And did that help you think it was noise?
I guess it did, yeah.
Yeah,
yes, it
okay.
did, but we know there's going to be noise anyway. But yes, I mean,
Of course.
model that. Yeah, I mean, what we can do with these kind of the computational methods, we can basically generate synthetic data. We can generate synthetic noise and run all the algorithms on that and see what happens. So we can help kind of fine tune our methods with that kind of approach.
Yeah,
there's a lot of great tools out there that have been developed by others that we can just pick them up and use them. That really helps.
Well, thank you very much.
Thank you.
Yeah. That was really good. Anyone else have any questions, or are we good? Go have lunch or dinner? Probably. All right, guys.
Yeah.
Well, thank you very much for doing this. Thanks to JR for coming and asking questions.
Well, actually, I have one more question that's not about your work. It's about our work. So as a patient advocacy group, can you give us any advice on what we should be doing? So we should just be, I mean, from my perspective, I think the SRF perspective in general is find as many patients as we can, figure out who they are, figure out what their variants are, figure out where we see missense, figure out all the, where we see VUS, try to see if we can figure out, especially with these single amino acid changes, right? Whether they're not sure what's happening, whether they're tolerated, whether they're pathogenic. And is there anything we can do besides try to find patients and make cell lines?
So there's one thing that, I mean, in this particular project, we were already working with two missense variants, and then the G344S was suggested to us because it's actually a very common one, which makes a lot of sense, but it's also that they all happen to be in the C2 domain. And so what we see here is that these variants, they're in the same domain, but still they are not obviously in places that influence the localization of the protein. So the C2 domain has previously been described as somehow important for unknown reasons to be promoting the gap activity. But no, we're not looking at the gap activity here. We're looking at the localization. So the way we've selected these missense variants is they're in the C2 domain. We just thought that would make sense I'm a biochemist by training, so we think about domains. But now if you think about the clinical side and the patient side, then obviously it's the symptoms of the important things, not really the domains and the residues. So what I can't see very easily, for example, if I look at the ClinVar data, it kind of said, yes, there are, there's symptoms here, or no, there are no symptoms here, but some kind of classification to link the missense variants to the symptoms would be really helpful. So for example, if we would look at different missense variants that appear to result in similar symptomology, that would be kind of useful.
Yeah.
So is that how, but I understand there, they're not that many. Yeah, well, there are many different missense variants and not that many patients who have one or other missense variant, but at least they could maybe be grouped in this way. Would that work, do you think?
Well, I mean, that's definitely a major goal for every, every pediatric geneticist, I think. But I think that what we're seeing in the patient population is that while 80% of them are protein truncating variants, they have a range of symptoms, and that the missense, the single amino acid changes that have been categorized as pathogenic, so those that we're considering missense, are mostly in domains that originally had a partial crystal structure, right? First off,
yes.
And that
Yeah.
so there's a little bit, so there's some that's not a I don't want to say biased, but the sample is, is not everyone. And then there's this basically the same range of symptoms that you see in the protein truncating variants. So what we're not seeing is, all the people with variants right in this region have the same symptoms and all of them. So it's more like there's a big range of symptoms that, everybody has some amount of all the symptoms, but some kids have way earlier seizure burden. Some kids have way more severe intellectual disability burden. Some kids have a much more difficult sleep burden. They can't sleep, but kids can sleep, right? So there's these different sort of silos of interrelated problems, because maybe the seizures are what's happening for the sleep, and maybe the seizures are happening for the ID, right? I mean, they're interrelated, but they're things that we kind of count differently. And we're not seeing really great patterns emerge, whereas on this slide, we see really great patterns emerge here, right? Like these are, we can tell the protein truncating from the missense right here. Absolutely. We're not really seeing that in the patient data, but I will say it is still somewhat early days, and there are people working on all the citizen data. So getting all our patient medical records put into a form that they can be digitally compared. Because when parents talk, when parents talk, it's really interesting. We're like, oh, my kid loves water. Oh, my kid loves water. Oh, my kid loves water, right? So we're kind of noticing some stuff. We're saying, oh, my kid can see really well in the dark. Oh, my kid can see really well in the dark. Oh, my kid can see really well in the dark, right? So parents have some interesting information that is really useful. But at the same time, we'll say, oh, my kid has a hard time with transitions. And oh, my kid has a hard time with transitions. Well, those can look completely different. One can be having huge amount, and first off, they're at different ages, right? But they can just be ultimately different classes, even if they have the same thing. So we have to have this sort of systematic way to understand. And then things like liking water and seeing well in the dark, those aren't really in our medical records. So anyway, I'm just sort of telling you the complexity. We would all love to see this play out a certain way, but it's not happening like it does in some diseases where the channel works more, over time, or it works not as much, right? Those are two different directions for a protein to go. We're not really seeing that yet with SynGAP.
So there's two things that come to mind there for me. So one is that, like I said, it would be quite nice to kind of do this kind of work with picking a bunch of, let's say, a bunch of missense that have some kind of similar symptoms, and another bunch that have other similar symptoms, but they are, two different groups. Do they fall into two different groups here or not, regardless of whether they're in the C2 domain or somewhere else? And then the other thing is what you said, that the different protein truncating variants are giving different symptoms, right? So
well, I'm
not-
saying they have a different profile. Like we're talking
Yeah,
about the
different
same
profile.
symptoms, but a different profile within
yeah,
that.
sorry, yeah.
Yeah,
That's what I mean.
yeah.
Sorry. But that means that the nonsense mediated decay is not working completely, right? Otherwise, there wouldn't be anything.
It's really not clear. It's just so not clear, right? Like it's really hard to say because it's hard. Like I think with the haploinsufficiency, since there is one gene working, with most genes in the human genome, one's enough, So there are fewer genes for which one, there's way more recessive diseases than dominant, right? Is that right? Or am I making that up?
I think so.
Yeah, so there is good protein in there. There's just not enough. And then the question is how much is the other protein just a disruptor or just irrelevant, right? Is the pathogenic variant irrelevant? Like it's just missing? And does it look just like a huge, multigenic deletion would look, where this, right? Or even just maybe not multigenic, but the full gene being deleted. Right. So there's nothing there to be a disruptor with a full gene deletion, right?
Yeah.
So we kind of want to compare that to everything else, but
so we don't know at the moment whether the difference in the profile of symptoms is because of some other genetic background effect, or because these truncation mutants are not actually being completely.
That's right. And I think
yeah.
I think what we're seeing though is that I'm not exactly sure why. I'm not exactly sure what the data is on this, but what I've heard and what I kind of instinctively believe is that SynGAP1 variants that are pathogenic have very high penetrance, and that they have a little bit of variable expressivity. So I think it's really high penetrance. And so I would think that perturbations in other genes, even just like even tolerated alleles in other genes in this background might have an effect, right?
Yeah.
So even if you had a SHANK3 or a DLG4, which we know there's a DLG4 binding site on SynGAP1. So potentially a change in DLG4 that's normally tolerated would be different in a SynGAP1 haploinsufficiency background, right?
Yeah.
That might uncover some change in another gene. And so there's a lot of genes that it can interact with. So, yeah, so I think it's just you and me talking at this point. So sorry to keep you so long. I obviously really I'm impressed with your work, but anyway.
But I have one more question. Can I ask you? So we know that there are at least 9 or 10 G344S patients.
Yeah. Right. So
how different is their profile?
Right. That's worldwide. I guess that's maybe that's like our strongest. Maybe that's our strongest help to you would be to go try to figure that out. They're not all in Citizen because I think a lot of them are other places in the world, and I'm not sure what language their records are in. But still, we could work hard on that.
Yeah. Interesting. Okay.
Thank you very much. I know Lauren had to leave. We're way past time, but and let me look in the chat. Let me just see. Oh yeah, that's Dr. Courtney. Thank you so much, Dr. Courtney. We are at SRF. We are so glad that you're doing this work. I'm so glad that you're so thoughtful about all the points of detail. I mean, it's just incredible. And we just can't wait to hear more from you and I think you have 9 people working on this, plus your finished supercomputers and all the other stuff. So thank you so much.
Thank you.